Okay, hello, and I hope everyone can hear me okay here. Welcome everyone to today's webinar by Randox Australia on lipoprotein a and apolipoproteins, reagents and quality control solutions. My name's Claudia, and joining me here today is Raj. We're both members of your local Randox Australia New Zealand team, and we thank you for your interest on this topic to be here live with us today, or maybe watching this back in the future. If you have any questions throughout the session, you're welcome to write them into the chat box, and then we'll try and get to them at the end. Or else we do have contact details on the last slide, and you're welcome to contact any one of us individually if you want to have a discussion. Okay, so I know many of you are probably familiar with Randox, but here's just a brief overview. So Randox is the UK's largest diagnostic company. We were established in 1982, and today we manufacture over 4.1 billion diagnostic tests annually, which we estimate to go on to diagnose over 370 million people annually. We played a large role in the COVID-19 pandemic. We facilitated over 26 million COVID-19 tests. But here today, we supply over 100,000 laboratories worldwide with a range of our diagnostic products. In terms of our global team, we have over 2,500 employees in offices or partner distributors across 145 countries, and 800 of us are dedicated R&D scientists. So today, the area of focus will be our niche reagent offering. So Randox offer a range of high-performance, unique and niche reagents that are designed to enhance the laboratory's testing capabilities. So our range of reagents are available for use on third-party auto-analyzers, and it allows the lab to expand their test menu without necessarily expanding your laboratory or introducing new instrumentation. So in that way, first of all, it can help reduce costs by perhaps bringing these tests in-house where you were sending them externally. And we also have a range of excellent stability and kit sizes available to help you reduce costs too. Our reagents are superior performance, so many of them are utilizing superior methodologies, gold standard methodologies where we can, or calibrated to well-characterized reference material. And all in all, this is to help assure confidence in our patient results. So as I mentioned, Randox offer many niche superior methodology reagents in several different clinical areas for disease identification or management, but today we'll be focusing only on our lipids portfolio. So particularly what we'll be talking about is lipoprotein a, known as the genetic marker for the risk of cardiovascular disease. We'll discuss small dense LDL, known as the most atherosclerotic type of any lipid. And Raj will then take us through a range of apolipoproteins in order to provide clinicians with a more advanced sort of lipid profile. So we'll start with lipoprotein a. Lipoprotein A, it's a lipoprotein complex. It was first identified in 1963, and its abundance in patient samples is largely determined by genetics. And it is estimated that 20 to 25% of people worldwide have an elevated lipoprotein A level. There's been various studies over the past few decades which have proven correlation between elevated lipoprotein A concentration and incidence of cardiovascular related events. So including this study here that has shown that those people with highest levels of lipoprotein A are at a 1.5 times increased risk of cardiovascular related death, 1.6 times increased risk of stroke, and up to 4 times risk of heart attack when compared with those at the lowest levels. There are now similar position statements on the topic globally, which recognise lipoprotein(a) as a risk factor for cardiovascular disease, and then some which go on to recommend screening for high-risk individuals. So this includes here from the Australian Atherosclerosis Society, which has stated that elevated lipoprotein(a) is an independent and causal risk factor for atherosclerotic cardiovascular disease and calcific aortic valve disease, and that selective screening strategies of high-risk patients are recommended. So the chemical structure of lipoprotein(a) is similar to that of LDL, but where it differs is the presence of this polymorphic glycoprotein, the apolipoprotein A. And this apolipoprotein A subunit has genetic variation across populations. So it's made up of subunits which contain unique Kringle domains. And typically, there is always one copy of this Kringle IV type 1, one copy of Kringle IV type 3 through to type 10, and one copy of Kringle type V. But where the polymorphism comes in is this Kringle IV type 2 domain here. So there's not always just one copy of this Kringle IV type 2. It's thought that there can be anywhere between 1 to 40 repeats of this particular domain in an individual. So this polymorphism means then that a person's lipoprotein(a) molecules can either have a relatively short ApoA subunit or a relatively quite long ApoA subunit, and this is where the challenges with measuring lipoprotein(a) can come in. So historically, lipoprotein(a) has been measured in mass units. However, as we can appreciate, simply measuring the total mass does not then account for the variability in the ApoA subunit size. So measuring in mass units can be misleading. It can overestimate lipoprotein(a) in those individuals with larger ApoA isoforms, or it can underestimate lipoprotein(a) in those individuals with smaller ApoA isoforms. And consequently, two people with the same mass concentration of lipoprotein(a) may have vastly different particle number and particle sizes, and that can lead to discrepancies in then how we manage risk or how we go on to manage potential disease. So consequently, molar measurement offers a more accurate reflection of the number of lipoprotein(a) particles present, irrespective of their size. So this is the more standardized and minimally biased method. Converting units is not always very straightforward, and in this case of lipoprotein(a), mentioned by various bodies on the topic, like the European Atherosclerosis Society, converting units is discouraged. So we can appreciate that the assay must report in nanomoles per litre to correctly identify the number of particles present rather than be skewed by the mass. But what about then the calibrator? You know, what do we do as manufacturers to ensure that the isoforms in the calibrator material is most appropriate? And what isoform is most appropriate to be using in calibrator material? Well, the answer is the mole, because essentially it is important that the assay can correctly identify all of the isoforms, hopefully, that are present in patient populations. The calibrated material with only one isoform of lipoprotein(a) does not reflect the heterogeneity present in the general population. And additionally, it is crucial that the calibrated material includes a variety of lipoprotein(a) isoforms at multiple points rather than single point calibration. And of course, it has to have accurately assigned molar values. So the next sort of challenge with measuring lipoprotein(a) then comes to traceability. Particularly considering the accelerating uptake of this test globally, it is important that we ensure all of our results are standardized, and manufacturers ensure that the reagents we're supplying are standardized. Fortunately, there is a globally accepted reference material available. This is the WHO IFCC reference material SRM2B. There is also an accepted gold standard test, and this is an ELISA from the Northwest Lipid Metabolism Diabetes Research Laboratory. They employ an isoform insensitive antibody, and it is meticulously calibrated with well-characterized material. However, this particular gold standard ELISA is not commercially available. So it is now accepted that methods based on the Denka-Sekan lipoprotein a assay are the most reliable commercially available lipoprotein a assays. In recognition of the various challenges and nuances of measuring lipoprotein a, there are various bodies around the world that have published guidelines for measurement of lipoprotein a, things that laboratories should consider when selecting their assay. So this particular guideline is from Heart UK, and this was published in 2019. And this is what it says here about lipoprotein a measurement. So they say serum lipoprotein a concentrations should be measured using a method where the effect of isoform size has been minimized using appropriate antibodies with calibrators certified for traceability of lipoprotein a values to the WHO IFCC reference material. Results should be expressed in nanomoles per litre of lipoprotein a particles. Conversion of mass units to molar units or vice versa introduces inaccuracy and should be discouraged. And currently, at the time of their publishing, only assays based on Denka reagents with calibrators traceable to nanomoles per litre to WHO IFCC reference material can be recommended. So this provides a good framework to be selecting lipoprotein a assays. How does this then fit into the Randox lipoprotein a assay? So just put here next to each point of the guideline how the lipoprotein a assay from Randox fits in. So first of all, our assay uses a large amount of the same isoform insensitive antibody as per the Denka and gold standard method. We also use a 5-point calibrator with heterogeneity that reflects the heterogeneity in the population. And this calibrator is traceable to the WHO IFCC reference material. Secondly, our assay is reportable in nanomoles per litre. We do not use any mass to molar conversion factors. And lastly, our reagent is based on the Denka-Sekan reagent with calibrators traceable to nanomoles per litre in nanomoles per litre to the WHO IFCC reference material. So here's some further information about the Randox lipoprotein a assay. We have applications available for a wide range of third-party clinical chemistry instruments, including these ones here on the screen, but this list is not exhaustive. It has a high tolerance for interference, so due to the wavelength at which it reads, it has a tolerance of up to 1,400 milligrams per deciliter of hemoglobin in samples, helping to reduce any issues with sample rejection. And there's 2 kit sizes available, so the smaller kit size and the larger kit size, which is about 3 times that of the small. The calibrator that we provide is purchased separately to the reagent. Again, it's a 5-point calibrator with that isoform heterogeneity that is really required. It's traceable to the globally recognized reference material. We offer instrument-specific set points for a range of common third-party clinical chemistry analyzers. It's all manufactured using 100% human serum for best commutability. And just here is the approximate values in nanomoles per litre of each of the 5 points of the calibrator to show that we have a quite linear spread of calibrated values. And Raj will talk a little bit later about the lipid QCs on offer from Randox, but I thought to mention this one here because Randox now offer a lipoprotein a elevated QC, which is completely standalone from our other QCs. And this is at the elevated level, which is now quite recognized around the world. Over 100 nanomoles per liter is the category of high risk. So it is important to have a quality control up around that level to know that you're correctly identifying the patient samples that come through. Okay, we'll move on now to small dense LDL cholesterol. So small dense LDLs are a type of LDL, but as the name suggests, they're smaller and heavier, meaning that they can permeate the inner arterial wall more readily. They're more susceptible to oxidation, and they assist in the deposition of fatty tissue, which is the beginning of the atherosclerosis pathway. Because of its small and dense size and structure, a study in the Journal of American Heart Association, only there in 2021, after looking at a variety of lipoproteins, concluded that small dense LDLs are the most atherosclerotic subtype of lipoproteins, and that optimizing small dense LDL levels may be paramount in atherosclerotic cardiovascular disease risk reduction. Traditionally, where we've just measured LDL, it just measures total LDL. We're not distinguishing between the different LDL subtypes, for example, small dense LDLs. But studies have shown there is a significant association between small dense LDLs and the development of cardiovascular disease, regardless of serum total LDL concentration. And this is really due to that highly atherosclerotic nature of the small dense LDLs. A person with elevated small dense LDLs has an increased risk of myocardial infarction by threefold, is what one of these studies had found. So Randox offer a small dense LDL reagent for use on third-party clinical chemistry systems. It has excellent correlation with the gold standard ultracentrifugation method, which is one of the only other methods available for small dense LDL, but it is very costly and very time and labor intensive. We use a two-step process, so two reagents. The first reagent will clear the non-small dense LDL lipoproteins, and in the second step, an enzymatic reaction occurs to identify the small dense LDL particles present. It has a wide measuring range from 4 to 100 milligrams per deciliter, which well covers the cutoff from our Randox IFU of 50 milligrams per deciliter. And again, there are applications available for a wide range of analyzers. Okay, so Raj will now discuss apolipoproteins in this next half.
Yes, thank you very much, Claudia, for the interesting first half of the presentation on Lp(a). In this second half of the presentation, I'll be going through apolipoproteins and the clinical significance of ApoA, ApoB, C2, C3, and ApoE. So to just start off with, apolipoproteins. So the question is, why apolipoproteins? Traditionally, when clinicians request for lipid panels, this would consist of cholesterol, HDL-C, triglycerides, and LDL. But this can only detect 20% of all coronary artery disease in patients. Hence why it's recommended that advanced lipids testing should be carried out to optimize patient treatment, both in the primary and secondary risk categories, and as such provide the necessary tools to present and reduce the risk of CVDs. And this is actually recommended by the National Lipid Association and the European Guidelines on Cardiovascular Disease. So what are apolipoproteins? Apolipoproteins are the proteins which bind to the lipids to form lipoproteins, and this actually makes it essential for these are essential for transporting lipids through the bloodstream. On the right hand side, you'll see a table with the apolipoproteins, the associated lipoproteins, and the major functions. They have various major functions, so depending on the apolipoprotein. And as we discuss each test in further detail, you'll realize that Randox is the only provider for the kits. So moving on, apolipoprotein. So it's commonly known as ApoA, and it's a signature apolipoprotein of the HDL and helps HDL particles remove excess cholesterol from the tissues. It plays a role in the reverse cholesterol transport through the activation of the lecithin cholesterol acetyltransferase pathway and the removal of free cholesterol from the extrahepatic tissues. ApoA may therefore be described as a non-atherogenic, showing an inverse relationship to cardiovascular risks. And actually, this means it does not promote any kind of formation of fatty plaques in the arteries, and hence why it shows an inverse relationship. So, higher the concentrations of ApoA, the better it helps to remove excess cholesterol from the bloodstream. In terms of the the patients with CAD generally displayed reduced levels of ApoA and increased level of ApoB, and we've seen this in recent studies where ApoA levels were significantly lower in stable coronary artery diseases and acute coronary syndrome groups compared to the control groups. And the normal range was indicated to be approximately 120 to 176 milligrams per deciliter. And interestingly, due to this, there have been recent studies where it's been explored how infusions of preparation of ApoA may actually improve a patient's cholesterol efflux capacity to reduce atherosclerosis. To move on into the Randox Apolipoprotein A kit, we have a wide range of applications available for a wide range of instruments. It's a liquid ready-to-use reagent with a wide measuring range of 6.50 to 233 milligrams per deciliter. It has a good sensitivity to be detecting deficiency in patients, and it's traceable to the WHO and IFCC reference material SP101. So moving on to apolipoprotein B, most commonly known as ApoB, it's the main form of protein found in the LDL and often correlates to CVD. Now, studies have actually shown that ApoB is in fact a good predictor of future atherosclerotic events, even when the total LDL cholesterol are within the normal or borderline range. Traditionally, we've seen that LDL is calculated using the Friedewald equation that has some limitations, such as the equation not holding true when the triglyceride levels are above a certain range of 400 milligrams per deciliter. For this very reason, we see that ApoB is actually a more superior test as they are less affected by triglycerides and intralipids, and unlike common LDL estimation methods. So the benefit of testing ApoB instead of LDL could be the fact that it takes into account all the atherogenic lipoprotein particles. This includes LDL and VLDL. Now, there's growing evidence that ApoB could actually replace the LDL marker, making a risk assessment more precise. But currently, ApoB is often tested alongside ApoA to determine the ApoB to ApoA ratio for CVD risk assessment. So, higher the ratio of the ApoB to ApoA ratio, the higher the risk of CVD. Randox offers a liquid ready-to-use reagent with a wide range of applications available for many clinical analyzers for ApoB. We have a wide measuring range of 11.2 to 184 milligrams per deciliter. And again, it's traceable to WHO and IFCC reference material SP307. Looking at ApoC2 and ApoC3 now. So ApoC2 is quite unique in the sense that it actually acts as a core factor for lipoprotein lipase enzyme. And this actually breaks down triglycerides in the lipoprotein into free fatty acids and glycerol. So ApoC2 is most commonly found in VLDL and chylomicrons. And it's important to actually note the clinical significance of measuring ApoC2. So although ApoC2 enhances the LPL activity, interestingly, some studies have actually found that elevated levels of ApoC2 are associated with excess triglyceride-rich particles that inhibit the LPL activity, which in fact increases the risk of CVD. but it is also important to detect the deficiencies in ApoC2 because there have been rare cases of decreased level of ApoC2, such as the hyperlipoproteinemia type IB, where there's hypertriglycerides level due to deficiency in the ApoC2 protein. So overall, ApoC2 actually closely mimics the LPL deficiency. So decreased in ApoC2 will actually correlate to LPL deficiency. Moving on to ApoC3 on the other side. Earlier I mentioned that ApoC2 actually activates the lipoprotein lipase. Now I'll talk about ApoC3, which will work completely opposite because ApoC3 actually inhibits the LPL activity, resulting in the delayed catabolism of triglyceride-rich lipoproteins. Yeah, so this means that there is an inverse relationship with the CVD where it comes to ApoC3 to C3. There's reduced levels of C3, which can actually increase the rate of triglyceride clearance from the plasma by up to 7-fold, as there is no inhibition of LPL activity. Hence, there is more efficient. There is more efficient breakdown of triglycerides. But elevated levels have also been associated with many conditions that increase the risk of CVD, such as type 2 diabetes, hyperbilirubinemia, kidney deficiency, and decreased thyroid functions. And all of these actually increase the risk of CVD. So the Randox Apolipoprotein C2 and C3 kits are liquid ready-to-use reagents. They have a measuring range of 1.48 to 9.70 milligrams per deciliter for ApoC2. So we want to make sure that we're measuring the depleted levels of ApoC2. And for ApoC3, we have a measuring range of 2.07 to 21.7 milligrams per deciliter. And this is also a liquid ready-to-use reagent. Moving on to apolipoprotein E. So ApoE is a multifunctional glycoprotein with central roles in lipid metabolism and transport, neurobiology, and neurodegenerative diseases. Interestingly, ApoE deficiency actually causes high serum cholesterol and triglyceride levels, leading to more premature atherosclerosis. Recent studies have also shown that ApoE is also involved in the transport of cholesterol in the brain, and deficiency or certain genetic variants of ApoE, such as the E4 allele, are associated with neurodegenerative diseases like the Alzheimer's disease. So the total concentration of ApoE can be quantified and assessed for deficiency or assessed against the proportion of ApoE isoforms for Alzheimer's disease risk. So the Apolipoprotein E kit is a liquid ready-to-use reagent with a measuring range of 1.04 to 12.3 milligrams per deciliter. It has an excellent precision of less than 2.79 CV, exceptional correlation of 1 when compared to other commercially available methods. Now, I'll just briefly look into the lipid controls and apolipoprotein controls. So the lipid controls are 100% human serum. They're lyophilized with a reconstituted stability of 7 days at 2 to 8 degrees or 4 weeks and minus 20 degrees. So they're true third-party control that Randox offer for ApoA, ApoB, and Lp(a). They have no sodium azide present, so no interference with clearance methods. We have 3 clinically relevant levels, which can all be purchased separately. We have 2 kit sizes available. It's a 3x1 or a 5x3 ml kit. Looking at the apolipoprotein controls, they're 100% human serum. They're lyophilized with reconstituted stability of 7 days at 2 to 8 degrees or 4 weeks at minus 20. It's a multi-analyte, so to consolidate down just to a small number of vials, we have 3 clinically relevant levels, which can all be purchased separately. Just below, we have the target values for each, depending on the method. And yep. Thank you very much for attending the webinar today. On this page, we have a list of representatives and their contact details that you can reach out to for any information. Please feel free to add any questions into the chat box and we'll do our best to answer your questions. If not, we'll get back to you.
Yeah, there doesn't seem to be any questions in the chat box, but we'll stay on for a couple of minutes if anyone has any questions. But if not, thanks very much for joining again, everyone, and we hope to see you again next time.